Okay, I have to say, got it here. Okay. So it's my real pleasure to welcome Stefan Heckers as our external speaker today. For disclosure, Stefan and I have known each other for many, many years, partly because of our common background in Europe, but also because he trained with Joe Coyle, who many of you know. But let me go back to very briefly, I promised, review his impressive background. So he is a native of Germany and went to medical school there. And in 1988, had magna cum laude, which is the highest level, got his MD degree, still in Germany. Then in 1990, he moved to the United States, has been here ever since. And first, he spent many years at Harvard, moving up the ranks. And just to be short, he became instructor, assistant associate professor in psychiatry at Harvard, and then was recruited at Vanderbilt in 2006, and is still since then chair of Department of Psychiatry. And also, since 2015, if I read this right, or 16, chair of the Department of Psychiatry and Behavioral Sciences, and that's important for you'll see what his focus is in research. He had many honors, 46, I counted them, major committee assignments in all the top-notch psychiatry-related organizations, institutions in the country and beyond. Associate editor of the Archives of General Psychiatry, for 3 years, editor-in-chief of JAMA Psychiatry. So you get the picture. He's a very, very special person, not only because of his background, but also because he is a really full-fledged clinician, but also a full-fledged basic researcher. And that's what makes it so attractive and so wonderful to read about his papers, talk to him, and as we hear now, listen to him. Stefan, it's all yours.
Thank you. Thank you very much, Robbie. One of my favorite people and one of my favorite institutions. And it's great to see Will there, who I've always kind of seen as the leader for schizophrenia research and still is. So I'm sharing with you my story, and it starts with a brief introduction. So I started at number one. I've spent some time in number two and currently am in number three. So I grew up in this very small hamlet at the Dutch-German border. And this is literally what I saw when I looked out of my bedroom window for many years. And this is what I'm seeing now. So there's still some water close by, but other than that, there are not too many commonalities between where I grew up and where I live now. And what brought me there, what brought me from Brüggen in Germany to Nashville, Tennessee? Curiosity. And this is also the common thread throughout the presentation. I really want to give first credit to a large number of people that have really supported me on this particular line of research. So these are all people who have really worked with me on hippocampal research in psychosis. It started already back in Germany. I've not listed them here, but then at Mass General, I got introduced to neuroimaging. At McLean, I continued imaging, but also went back to my postmortem roots. And at Vanderbilt, I've been primarily interested in neuroimaging studies. And you can see some collaborators, leaders really in memory research and funding. But the person that I really want to highlight first is mentioned twice, and that is Christina Conradi, my spouse and collaborator for many years, who's really helped me make many of the contributions on the postmortem side, introduced me to molecular techniques such as in situ hybridization and others. And you can see here that we didn't have the pitchfork when we took the photo, but it is somewhat an American love story because I wouldn't be in the United States without her. And I clearly wouldn't be here anymore without her. All right, switching gears. The hippocampus is a small brain region in the medial temporal lobe. When I use my cursor, can you see this on your screen? You can
Yes,
see
that works. Yeah.
Okay, so this is the parahippocampal gyrus. So this is the left hemisphere of a human brain. This is looking at it from the convexity. All of the white matter is taken away, and you can see the full extent of the hippocampus. A cut through the hippocampus at that level, number 2, would give you a Nissl stain section like this, or an MRI image looking at the structure and also activation patterns. So Christine and I have been interested in this topic of abnormal circuitry in the hippocampus. So you have 3 levels of investigation. You have macroscopic abnormalities such as volume of the hippocampus or shape of the hippocampus. At the more cellular level, you have the trisynaptic pathway of the hippocampus and the connections that bring information into the hippocampus. So very briefly, for those who are not hippocampal aficionados, the entorhinal cortex receives sensory information. That sensory information is directly sent to the CA1 region, it's so-called direct pathway. In addition to the sensory information, previously stored, previously recorded and stored information can be retrieved through the indirect pathway. So it also arrives at the entorhinal cortex, but then goes through this trisynaptic pathway and then arrives at the same neuron, the pyramidal cell in area CA1, coronary ammonis sector 1. And it is this, you can see here the blue and the red axon terminating on the same dendrite. It is that coincidence detection that allows the hippocampus to do the one thing it does, which is separating old and new, to be a detector of previously stored or novel information. And it is our hypothesis that that disintegration, that mislabeling of new and old information generates some of the features of psychosis, delusions and hallucinations, the inability to separate internally created representation from online sensory data. And it is also tied to some of the cognitive deficits, primarily the associative memory deficits. Well, what is the evidence for this? Well, it turns out that people who have looked at the brain for more than 100 years, in fact, Kraepelin already looked at the brain. He had his assistant, whose name was Alois Alzheimer, do the initial studies of postmortem research in schizophrenia. Few people know that Alzheimer devoted most of his career to psychosis research and only published one single case at a summer meeting on a daily age. And from there, Alzheimer's disease was created. But in 1985, for the first time, a researcher proposed that it might make sense to look at the hippocampus. Until then, for almost 100 years, schizophrenia research had literally ignored the hippocampus, which is part and parcel of the story of the hippocampus in neuroscience, because until patient HM, until Brenda Milner and others really focused on the hippocampus as an important brain region for memory, it was also ignored in neuroscience in general. But for those who really like scatter plots and real data rather than just nice fancy data, look at this. So this is the hippocampus in Bogart's original paper. That's the first paper that really showed abnormalities in the hippocampus. This is the individuals that are considered healthy. And this is the group of individuals with, at that time, was called dementia praecox. This was a very old brain collection. You can see that there is quite a bit of scatter, and that is a recurring theme. So it's not completely non-overlapping. Even in this, which is a 31% volume difference in the hippocampus, even in those rather striking group differences, you find significant overlap. But now, having done many, many structural MRI studies, so this is one of the many meta-analyses looking at region of interest ROI studies, not VBM, voxel-based morphometry studies. So this is a better way to really look at hippocampal volume. You can see here in this red box, these are decent effect sizes of 0.4, 0.5, 0.6. In fact, the hippocampal volume difference is the most robust of all brain structural changes in schizophrenia. In this paper from Christos Pantelis and Dennis Villakoulis from the Melbourne group is really important because it shows clearly what I just showed you. The chronic as well as first episode patients already have smaller hippocampi, typically hovering around the 5 to 10% volume difference range. But their contribution was that there seems to be a gradient, there seems to be an accentuation of that difference. with less pronounced differences in schizophreniform patients. So for those who don't know the DSM well enough, that means people with a psychotic disorder that has run between 1 and 6 months. So it's a schizophrenia in the making, and has many of the clinical features, but it's clearly the early stages. So this was a clearly important paper leading many of our research studies with a simple question: when does hippocampal volume occur, a volume change occur, and does it change over time? So since I was trained originally as an anatomist, I need to get started on the true anatomy. That's what the hippocampus really looks like. So if somebody tells you they have a 7T scanner and they can show you beautiful images of the hippocampus, just laugh them off, because they have no idea what the hippocampus really looks like. It's a very small brain region. It's about 3 to 4 cubic centimeters. It's less than 1% of your entire brain volume. So on the left upper corner, you see the Nissl stain. Even with microscopic details of the Nissl stain, you cannot distinguish the 4 subfields of cornu ammonis. You need additional preparations such as Calvindin stains or palpabumin stains. And only with those can you then see the very sharp demarcation of, for example, CA1 and CA2. And it's that very detailed anatomy that then allows you to do a parcellation of the full extent of the human hippocampus that then shows you 5 different layers. Now, there are people who are publishing papers that in 7T images, only the stratum oriens or only the radiatum lacunosum moleculare, layers 5, 4, and 3 here, are different. I have looked at 7T images, and it is not possible these days to make the separation of the human hippocampus work out in imaging data. You need the anatomy. And I want to make this very clear, because we make some statements about CA1 being particularly affected, but we have to be humble that there's a huge gap between the postmortem research and the neuroimaging research for the study of the human hippocampus. Now, I've spent a lot of time in the morgue. This is what a real human brain looks like. I hope it doesn't scare people. So this is what, when you still, it's not even paraffin, it's not even formalin fixed. This is truly a normal brain after about 2 days or so after having transported to the brain bank. And I spent a lot of time dissecting those and taking those pictures. And the line here, the blue line, is an important one, because that is the distinction between the anterior hippocampus, often called the uncus, Greek for hook, because that's what the hippocampal shape looks like here, versus the body and the tail, which is this part. So I'll come back to this anterior-posterior distinction because it's functionally very, very relevant. It really started with Brian Strange publishing a really important review paper in 2014. It really has taken off from here. Researchers have spent much more time now to understand the anterior-posterior gradients, because until then, it was all Cajal and others who in the 19th century had identified the subfields, the dentate gyrus, the cornu ammonis, the subiculum. So we had this idea that it's the laminar or lamellar architecture of the hippocampus that derives the function. But we now know that also the anterior-posterior gradient are absolutely crucial for understanding the role of the hippocampus in disease. So we have embraced this segmentation in our imaging data, but it really requires a lot of detail. So you can look up Maureen's paper from 2018, where she outlines what exactly we mean when we talk about the anterior and the posterior part of the hippocampus. FreeSurfer, which is considered the gold standard, cannot do that for you, because FreeSurfer doesn't have the anatomical parcellation that allows you to group correctly together the cornu ammonis subfields, the dentate gyrus, and the subiculum. So using this parcellation, we could show a couple of years ago that the anterior hippocampus, the head of the hippocampus, is smaller in both chronic patients as well as patients in the early stages of psychosis. The early stages of psychosis in our studies going forward for the rest of the talk means patients in the first 2 years of a schizophrenia spectrum disorder. That's number one. And most importantly, the majority of those are within the first 6 months. So that means schizophreniform disorder. So there was an early indication that the anterior hippocampus might be more affected than the posterior in the early stages. Whether there's progression, we do not know. I will show you some data on this, but it's clear that there is some difference between the early stages and the chronic stages. So this is taken from our most recent R1 renewal. So we have now, for over 10 years, studied the trajectory of illness, studying early stage of psychosis individuals and follow them for 2 years. And I have to acknowledge, although Will clearly could have told me that before I started doing this, that a good number of patients with schizophreniform disorder do not progress. And we found that it's a 1/3, 2/3 split in our sample, that out of 100 first episode patients with schizophreniform disorder, about 2/3 progress to schizophrenia over 6 months. In fact, we follow them for 2 years and more. So we know the progression actually after 2 years is about 2/3. But 1/3, even after 2 years, have not progressed to schizophrenia. And we are now learning a lot about the differences between those two groups. And then in a renewal of the grant, we are now following this first cohort 5 to 10 years into the illness. And we are now also separating within the schizophrenia. They all have progressed to schizophrenia, but we have good and poor social functioning. And then we can separate those. This is something that we are still collecting data on, but I can show you a couple of exciting data on that first wave. And the way that we do this is, so this is kind of my obsessive German nature. This is all 63 early stage psychosis individuals mapped out. Each person is one line. Everybody starts at time point 0. That means the onset of psychosis. So we have people who have been literally recruited the week that they became for the first time psychotic, versus the last person who was still within the 2-year window. You can see that that's how we defined our cohort. So the initial data point is always within 2 years of onset of psychosis, but then each dot indicates an MRI scan. So you can see that for the majority of those 63 individuals, we've actually had multiple, sometimes 4 scans over a 2-year period. And then the color indicates whether they stay within the same diagnosis or whether there's conversion. So let's take this person all the way at the bottom, 3 MRI scans over the period of 2 years. And at all 3 time points, the assessment confirmed that it was still schizophreniform disorder, meaning no conversion. Compare that to those who turn from red to blue. Those are all of the converters who started out as schizophreniform, but then at the end of 2 years get the diagnosis schizophrenia. So what happens to the hippocampus? So these are the spaghetti plots. Again, I'm detail-oriented. This shows you each and every case over a 2-year period. Each dot for those lines shows you the MRI scan at that time point. And you can see the mean, and you can see that these are literally flat lines. Blue indicates healthy individuals. So in addition to those 63 patients with schizophrenia spectrum disorder, we also had a similarly large healthy control group without psychotic disorder, without mental illness of any kind. And you can see that these are flat lines. There's quite a bit of variability, but over a 2-year period, there are flat lines. But that is now allowing us to test the main hypothesis. And this is the surprising finding. Even at time point 1, meaning when people have just started having psychotic symptoms or are in the early stages of psychosis, the group in blue is the healthy control group. And this group with the circles, the round circles, well, all circles are round, have a similar hippocampal volume like the healthy controls. And both of those are statistically significantly different from those who start out with schizophrenia, or, very importantly, who start out as schizophreniform but then progress to schizophrenia. So it is a hippocampal volume difference that is very significant, particularly in the anterior hippocampus, not in the posterior hippocampus, that separates in the early stages of psychosis those individuals that do not progress to schizophrenia, who have normal hippocampal volume, versus those that do progress.
Stefan, can I ask a quick question?
Yep.
A couple slides back, you had called the people who didn't progress SF. What does that stand for?
Schizophreniform.
So those people basically
schizophreniform over a 2-year period means people who get assessed with PANS and full skids every 6 months and have met the criteria for schizophrenia criterion A, but do not meet the time criterion. So
Okay.
relapse.
They
And
didn't get followed after 2 years?
and we are following them now for 5 years, but the data is from the 2-year period. Yeah.
Okay.
All right. So
Is
that's
it the time criteria and also the level of functioning criteria too? Because I had the same question and then I just pulled the DSM because I'm not a psychiatrist, but
well, these are two different. So you need both. You need all three. First, you need to meet criteria A for schizophrenia. You need 2 out of 5 criteria A. They all meet that. Secondly, they need to meet the criteria for impairment. Well, that's a tricky one for schizophreniform disorder because that's only a required criterion for schizophrenia, not for schizophreniform disorder. But
Yeah.
our schizophreniform disorder patients also meet that criterion. They're literally all our hospitalized patients. And then the third is you need to have symptoms for at least 6 months with functional decline and active symptoms for at least 1 out of those 6 months. And that is not reached by the schizophreniform patients who don't progress.
okay.
So they are same on 2, but they're lacking on the 3rd. So that's one exciting finding. There's another evolving story that we just became aware of. So I hope that comes through. So this is what a human hippocampus looks like with MRI scans. This is a 7T scan. Okay, and people tell you that they can subdivide the CA sectors on those images. I just rest my case. But look at this here at the top. This is what the development of the hippocampus actually goes through. This is the cortical plate in the embryo in the first couple of weeks. And out of this sheer or out of this simple layer of cortical cells in the medial temporal lobe, the hippocampus is formed. So this is in the 2nd trimester, at the end of the 2nd trimester, what the hippocampus looks like. It doesn't really change in the 3rd trimester very much, but that's what the adult hippocampus then looks like. And we now have learned a couple of years ago that there are individuals who have an incomplete inversion of the hippocampus. So they literally have a hippocampus that looks like this and not like this. And you can see here, this is an example of a incomplete hippocampal inversion, IHI, and this is the normal hippocampal shape, where the hippocampus is flat and where the subiculum and the head of the hippocampus are further apart. So you can now take quantitative criteria, there are actually 5 different criteria, all indicated by those lines, that now allow you to quantitatively assess how far away each hippocampus is from the normal inverted hippocampus. So this was originally described in epilepsy. Epilepsy is the one neurological disorder that is tied to significantly increased incomplete hippocampal inversion. And this is what it looks like in schizophrenia. These are the distribution plots on the left and on the right. IHI is much more prevalent on the left. It also explains now, finally, why the right hippocampus is substantially larger in the human brain, because the hippocampal inversion is much more complete on the right. Only 4 out of the healthy and 10 out of the patients percentage had incomplete hippocampal inversion. But on the left, 15 of the healthy population and 27 of the schizophrenia group had incomplete hippocampal inversion. This explains to a large degree the hippocampal volume differences. If that is true and can be confirmed, again, we are the first to report this, this would be for the first time a clear indication of a neurodevelopment mechanism of hippocampal volume difference in schizophrenia. Just think about that. It gets even better, because once you know that, you can not only look at the volume, you also look at the shape. So this is an MRI scan. Somebody sits there like Max Borowski, the MD PhD student who just finished his medical school and will become a psychiatrist, thank God. He has spent hours and hours to do the segmentation of these hippocampi, and they now look like this. They look pretty ugly, because these are the voxels on a 3T scan that make the hippocampus look not very smooth. Then you can take, however, these SPALM-D tools, these neuroimaging analysis platforms that allow you for the individual hippocampus to do a spherical model of the hippocampus. These spherical models can then identify a particular location of the hippocampus in its native space. And then then you get images like this. So this is one hippocampus, but it's now smooth, taking into consideration the shape. And what you can then do, these vertices of a particular shape can then be analyzed statistically. And you can then map out for a large group. So we had about 300 individuals, 300 patients with schizophrenia that we studied, to then look at statistical differences in the shape. And that's what the statistical differences will look like. Any of the areas that are in blue are more concave, and areas in red are more convex in patients with schizophrenia compared to healthy controls. And when we did that in the sample, we found this. So this is an analysis of 300 patients and 300 healthy controls, looking at the hippocampal shape, and these areas in blue indicate very significant differences between schizophrenia and healthy controls. This is something that was well known. It was published 20 years ago, has been confirmed many times that the region of the hippocampus here, the left lateral convexity, is particularly affected in schizophrenia. This model does not include the IHI information. But if we now include the IHI information in our shape analysis, stay focused on this area here, it completely disappears. So the well-known shape differences in the anterior hippocampus that have been described for 20 years are in large part due to this new developmental mechanism of an incomplete hippocampal inversion. So that's the difference with and without the IHI information. And you can then even take the information and say, I can now superimpose a subfield segmentation of the hippocampus and can show that the differences that we identified that are due to IHI are entirely in CA1, in anterior CA1. And from now on, for the rest of the talk, that's where the focus will be for all functional changes. All of our CBV imaging, all of our bold signal changes are entirely in anterior CA1. There's something very special about that brain region versus the other subfields and regions of the hippocampus. However, having said that, there's also great variability. So this is one individual, and you can see that there are various sectors of CA1 that will show up in different parts of these coronal sections. So somebody who doesn't really know where CA1 is can easily get confused and say, well, that looks like the normal hippocampus. So that's true, but that's the easy part. This is the hard one. That's the ANCAS, where you might even have 3 cuts through CA1, all interspersed with the CA2, CA3 sector. All right, you can see that I'm getting very excited about, boring human neuroanatomy, but it never stops to, it just amazes me how the human brain is organized, even a simple structure, a 3-layered structure like the hippocampus. Okay, so I will spend a couple of minutes on the postmortem research. I'm not going to spend too much on this. So again, Christina has helped me greatly over the years to do some of those studies. And we were one of the first to really show that overall hippocampal cell number is not really that different between patients with schizophrenia and healthy controls. It's not a neurodegenerative illness. So this gives you the total cell count. You can see here about 10 million cells. So that's what the human hippocampus has, about 10 to 11 million cells per hippocampus. So 20, 25 million total for the human brain. That's it. Considering that there are 10 to the power of 11 or 10 to the power of 12 neurons, it's a very small number. We now know from a meta-analysis that Max Walrowski did and published a couple of years ago that CA1 seems to be also a hotspot for our findings for both volume as well as total cell number changes. So there seems to be something about CA1 and most likely more the left than the right that was always puzzling to me until I learned about the IHI differences. Again, the left hippocampus development lags behind by several days. And imagine a neurodevelopmental hit that is particularly targeting the second trimester will impact the left hippocampus much more so than the right hippocampus, simply because the inversion of the hippocampus and the outgrowth of cells into the pyramidal cell layer is much later in the left than in the right. So we spent quite a bit of time looking at each and every cell. So this is from a 50 micron section. Each of the colored dots is a pyramidal cell. Each of the white circles is an interneuron. You can just toggle back and forth. And if you count those, the ratio of pyramidal cells, glutamatergic cells to inhibitory interneurons, GABAergic cells is 10:1. There's no other brain region in the human brain that has as much of a lopsided excitation over inhibition ratio. In the cerebral cortex, it's 3:1. In the thalamus, it's also about 3 to 4:1. So we spent a lot of time looking at these. So this was our original study that we did when we were still back at McLean and working with Francine. This was the first in situ hybridization study of GAD65 and GAD67 in the human hippocampus in schizophrenia. And we showed differences in both bipolar disorder as well as schizophrenia. But then we took it one step further and said, well, interneurons are not just interneurons. So I told you about these pyramidal cells. They ultimately are the drivers of the output, but they are surrounded by all of these local interneurons. Some of them are targeting the dendrites, some of them are targeting the soma, some of them are targeting the axon hillock, but some interneurons target other interneurons, so they are actually disinhibitory. So we need to actually understand this. So we used both protein as well as gene expression to subdivide those. So then that's how we got all of these beautiful stains. Not only were they able to allow us to separate the subfields, but we were also able to count the neurons. And we found rather striking differences for two subpopulations, somatostatin and palbalbumin. Those are the two subtypes of interneurons that are reduced by 40% in total cell number in the hippocampus, and particularly affecting CA1. All right. So now the plot is thickening. So now we have volume differences being the most robust for all brain structural differences in schizophrenia. We have a developmental mechanism that could be IHI or others, and we have a clear indication that a person who progresses to schizophrenia will look different from a person who has a full-fledged psychotic syndrome, but who will not progress to schizophrenia. Now that we have all of that, we need to develop a model. And so Christina, with her artistic skills, helped me with this. So this is the pyramidal cell, and these are the two different types of interneurons that modulate the firing or the activity of this pyramidal cell. They actually are localized in different layers. They're also preferentially expressed in different subfields of the hippocampus. And this is very similar, this graph, but this is the anatomical detail that we were able to contribute, is very similar to John Listman's Trends in Neuroscience paper, which was really a final product from Joe Coyle's Conte Center that was focusing on NMDA receptor mechanisms in psychosis. So the story here is that these GABAergic interneurons, and again, these are the palbalbumin fast spiking interneurons, not anything else, not calretinin and not calbindin, but palbalbumin. They are the ones who provide the negative feedback loop from this axon collateral through an NMDA mechanism, but then ultimately an expression of GAD65, 67, an expression of these proteins that we've identified, these calcium binding proteins. So this machinery is not working properly and therefore leads to an increased readout and increased firing from those pyramidal cells in target regions. So this is how we've conceptualized using neuroimaging. So if that is really at the core of the hippocampal problems, that the pyramidal cells are hyperactive because the interneuron does not provide the negative feedback loop, the pyramidal cell firing is increased, the release of glutamate into the hippocampus is increased, it leads then to other changes in either glial cells or other neurons in other regions of either the brain or the hippocampus, and that can lead to changes in either cerebral blood volume or cerebral blood flow. Now, these are speculations because we don't really know exactly how disinhibition leads via increased glutamate release to those changes in CBB and CBF. It's quite challenging to make claims. So I need to make that leap to actually use neuroimaging to study this. So I'm giving you a brief review of what we have learned over this, first with PET and then with MR technology. The original paper is actually a paper that Peter Little and Carl Friston published in 1992. This is the first paper showing left temporal lobe hyperactivity in schizophrenia, not in all patients with schizophrenia, but in those with the reality distortion syndrome. So you don't really find it in the psychomotor poverty syndrome. That's the British version of the negative syndrome. Also not in the disorganization syndrome. So it would be the hebephrenia subtype, but it is in the reality distortion syndrome. So those with prominent delusions and hallucinations, they have a significantly increased O15 blood flow. And this is a technique that was able to measure absolute blood flow because they did arterial blood sampling. For those who really care about neuroimaging, this is the paper for which Carl Friston wrote a program to analyze the data. And this is the first paper that used a program called SPM to analyze PET data. And Carl Friston literally wrote that program simply to analyze this data set. Just a little historical tidbit. We had a similar finding a couple of years later, also with PET, but we did this under the constrained condition of a memory task. So we looked at hippocampal blood flow modulation during a performance of a memory task. And in healthy individuals, we got a significant increase in hippocampal blood flow that we could identify even at the individual subject level and correlate with accuracy of memory recall. That was completely lacking in patients. There was no increase in test, there was a decrease, and there was overall, across all three conditions, a significantly increased baseline. So that became our story, to study hippocampal hyperactivity and the lack of recruitment during task performance as a consequence of disinhibition in the hippocampus. Now, there are many different techniques that people can use with MR to actually study activity and recruitment. We identified two that we focused on, and I will show you primarily results with those two techniques, although we have others as well. But those are the ones that I can focus on today. The first would be the steady state analysis of cerebral blood volume using gadolinium. Very rarely done. In fact, in my awareness, there are only two groups that have ever used CBV imaging in schizophrenia, and I will show you those results. And then, of course, the majority of people use functional MRI and the Bolt signal. So we built on this idea that there's hyperactivity and inability to modulate. This is one of the examples in early study, very boring presentation of visual stimuli faces, and then simply measuring hippocampal Bolt signal over a period of 100 milliseconds or 80 milliseconds. And you could see that there is a pattern in healthy controls in these open squares, where the first half of the experiment, you see those faces. You actually are still curious, you're paying attention, but then you habituate over time. And that early curiosity sign is there in patients with schizophrenia, but it remains. And the person does not habituate, the hippocampus does not habituate to previously stored information. We did this in much fancier versions where we not just looked at one visual stimulus, but different types of faces, smaller ones and larger ones, objects, different kinds of objects. So we did all of these very nice permutation of the design to tease apart the novelty signal and the habituation signal, and we could show that. So this is the healthy control group in blue, and this is the patients with schizophrenia. This is the signal when you look at repeated stimuli, habituation over time, and this is the hippocampal signal when you look at novel stimuli, either faces or objects. And that separation, that delta, the difference between habituation to repeated stimuli and the increased hippocampal signal to novel stimuli is completely lacking in patients with schizophrenia. We use that in more recent studies to then make a tie between this lack of habituation and tied this also to the performance in the memory tasks. So we hypothesized that if there really is this lack of habituation, this could also then impair relational memory. And so we did this looking at this very long experiment and looking at the early detection of either a face or an object, And you could see that that signal is related to the ability to recognize a stimulus and that was previously learned. And that was from a data set. I cannot give you the details of that. It's the ability to relationally bind a face to a scene, and the ability of the person to identify which of the three target faces is the right one that was previously shown on the background scene. And so this impairment in relational memory, that we have shown many times, which is one of the most robust cognitive deficits that we found in our patients, is also related to an abnormal modulation of that signal in the MRI data. So with better relational memory, so healthy controls are in blue and early psychosis are in red. The healthy controls show a negative correlation between relational memory and habituation. And that means the better relational memory is related to less habituation of the signal. And that is completely lacking in patients with schizophrenia. All right, going back to the imaging, so I leave kind of the whole memory story out for today. We've really tried hard to find the signal that indicates hyperactivity as a readout of increased glutamate and decreased interneuron function. So we started out with CBV, and if we could do CBV in many patients, we most likely would do that going forward as our primary technique, because it has been very reliable and it also been very consistent over different cohorts. However, it is a very challenging technique to use in patients, and it is particularly difficult to use with repeated study design when you want to study changes over time or the effects of treatment. So Scott Small, a neurologist at Columbia, really is a pioneer of this study of cerebral blood volume mapping. Here he's showing you the relationship between PET fluorodeoxyglucose as the best measure of overall metabolism or overall tonic activity, and how that is related to the CBV signal. So he has shown very clearly that CBV might be the best readout next to PET FDG for tonic brain activity or metabolism. He showed in the 2009 paper, with Scott Schobel being the first author, that CBV is increased, and it's particularly increased in CA1. This is actually coming out here in the heat map as this particular region being different between healthy controls in patients with schizophrenia. And more importantly, he showed in a follow-up study in 2013, again, the hyperactivity here in CA1. And if you now look back at the images that I showed you with 7T, you have to look really, really hard to even see the hippocampus, right? So these are 3T images. Superimposed are the cerebral blood volume maps, which of course don't have any spatial information about the hippocampus. They are just CBV maps. And then get this. There are 6 slices through the hippocampus from the anterior, the uncus, all the way through the back and the tail of the hippocampus. Only one of the 6 sections of the hippocampus shows a difference in cerebral blood volume mapping. So the entire difference between the two groups is based on one single slice through CA1. So we tried to replicate this. So we did a similar study. Very high resolution. This is a 1 millimeter isotropic voxel technique. So it actually gives you the ability to identify individual voxels that then can be labeled as subiculum, C1, C23, dentate gyrus. And we found, get this, also a difference, but it was only in the anterior part of CA1. We find it actually in 2 different slices, in slice 2 and slice 4. Slice 1 typically is just anterior to the hippocampus. And we don't find it in the other regions. In fact, in CA3, you might even find the opposite pattern, where there is a decreased cerebral blood volume measure in the anterior part of CA2, CA3. There also are tremendous gradients which are actually quite interesting because the subiculum is the primary readout of the hippocampus. So we published on this separately. So this is a really nice technique to give you high resolution, subfield specific information. But I just want to caution people who want to do this kind of imaging. There is a signal there. There are only 2 groups who've ever used it. Both of them have found the same thing in several cohorts, but it is literally one slice through the hippocampus. All right, now I switch to BOLD, and we have many data sets. I'm just going to show you the one that Maureen published a couple of years ago in the American Journal. So this was a nice study because we used a very simple functional MRI paradigm where an individual lying in the scanner simply is passively viewing either scenes or faces or scrambled images. And other groups had previously shown that the passive viewing of scenes versus the passive viewing of either faces or a scrambled condition allows you to show significant differences in hippocampal BOLD signal. So there seems to be something special about the viewing of scenes. Of course, this has been tied to the role of the hippocampus in spatial memory and exploration of spaces. There are several theories why that is, but we found very robust activation. You can see here that this maps shows you how many of the individuals in the study had an overlap in the activation in each voxel of the hippocampus. So for people who do hippocampal imaging, it's very rare to find robust activation patterns. This is by far the most robust I've ever seen in 20 years of hippocampal imaging. You show it also in patients with schizophrenia or early psychosis, but there's a significant difference between those two, and that's shown here. However, in this data set, we also had a subset who had the CBV imaging. So we now had the ability to look at, okay, how is cerebral blood volume mapping, where you previously showed hyperactivity, how is that related to the ability to recruit the hippocampus during task performance? And that's where we found this really interesting relationship. Again, healthy controls show no relationship. So the activity at baseline or the tonic activity measured by CBV does not predict the percent signal change during task performance. But in patients with early psychosis, again, the first 2 years of a schizophrenia spectrum disorder, there you find a significant difference with those who have the hyperactive hippocampus having a decreased ability to recruit the hippocampus during task performance, which of course goes back to the original PET paper that I showed you from the 1990s. So we are pursuing this, we are continuing to do this, and it is really a work in progress. We are at this point about halfway through collecting a new cohort of early stage psychosis or early stage psychosis patients. Again, that's the yellow area here. We are targeting another 100 individuals. We expect that, like before, we have about a 1/3, 2/3 split in either converters or non-converters from schizophreniform to schizophrenia, bolstering our case that there will be differences already early on that allow us to separate patients who progress to schizophrenia and separate them from similarly ill individuals with psychosis who do meet all criteria for schizophreniform disorder but don't progress. But of course, we also then have our original cohort that we can then follow to see whether there are any further changes 5 to 10 years into the illness. So we are trying to find kind of an epidemiological study design. So the Suffolk County cohort, for example, where people have been followed after an initial episode of psychosis and then followed over 10, 20 years. We are trying to use a similar study design for our first episode cohort. Challenging to do, but we worked very hard to get the clinical infrastructure in place to do that. And one of the benefits of living in the South is that people actually are not as much moving around from one place to another. And our attrition rate is less than 20% over a 5-year period for patients. In fact, our attrition rate is more of a problem for our healthy control participants because they're moving around more. So it's one of the real assets that we have, that we are building good connections to our patients. Many of those are also in our clinics. And so we are able to keep them coming back for clinical assessments and neuroimaging. The last thing I want to tell you is, unfortunately, I cannot share you the data because we are literally in the process of analyzing them. But we've done one more thing, which is testing the hypothesis with an intervention. So if it's true that the hippocampus is hyperactive, and if we also believe that this is already a pattern that is there early on because of a new developmental mechanism, what can we do either pharmacologically or with other means to decrease hippocampal hyperactivity? And does that then lead to changes in either cognition or clinical performance? So at the top here, you see a simple diagram that shows you the various targets in the hippocampus that we could identify for a modulation of activity. So you have GABA A receptors, so think about benzodiazepines, for example. You have glutamatergic neurons that are targeting on GABAergic cells. Some of those are modulated by CB1, cannabinoid 1 receptors. There are alpha-7 nicotinic receptors, primarily modulating these glutamatergic cells in the hippocampus. We have focused on a mechanism that doesn't use a receptor like the alpha-7 nicotinic receptor, the GABA receptor, or the CB1 receptor. We focused on a potassium or an ion channel that is regulated by the anti-epileptic levetiracetam, also called Keppra. So very well known to be effective in the treatment of complex partial seizures, very well known to target the hippocampus and stabilizing excitation inhibition balance in the hippocampus. And so what we have done, also Maxwell Roski, who has done quite a bit of work over the last couple of years, pioneered this for us. We recruited during the pandemic 60 individuals, 30 with a diagnosis of schizophrenia. So these were not early stage psychosis patients. So these were more chronic patients, and then matched them with 30 healthy controls and had them studied with an MRI scan twice. And under one condition, they were given a placebo pill, and 2 weeks or 1 week apart in the other condition, they were given one dose, a single dose of 500 milligrams of levetiracetam. So there are other studies underway. In fact, I'm aware of at least 2 other investigators who are using levetiracetam for the either treatment of clinical symptoms in schizophrenia or for the modulation of hippocampal hyperactivity. So we are not the only ones who are doing this, but I'm not aware of any data from those studies. There's a clinical study that was just published last year from Iran that has published some clinical data, but to my knowledge, there are no imaging data so far published. And I would like to tell you a bit more about this, but we even wrote a manuscript and then realized that we have some other data that we haven't even looked at. And so the story is emerging, and we are hopeful that we can show with this very simple study design 2 things. First, that at baseline during the placebo condition, we find increased hippocampal activity like we've always found it with our imaging measures. And that then secondly, we find either a main effect of treatment, meaning levetiracetam will normalize hippocampal activity, or even, the best case scenario, a group by treatment interaction. Since the activity of the hippocampus is substantially increased in patients, we anticipate that the levetiracetam effect will be greater in the patients than in the healthy control participants. So that kind of wraps up my slides. So that gave you a fast review of 25 years or so of looking at the hippocampus, but I hope that you agree that it does make sense to look at hippocampal structure. Our newer findings of a new developmental mechanism and of a very specific deficit, structural deficit in CA1, I think really is helpful because it's not just the hippocampus overall, it is the anterior CA1 region. And I think that with more effort being paid to capture hippocampal activity with imaging, we can take this method of looking at hippocampal activity into clinical scenarios and also study the effects of treatment, and then hopefully also can make a stronger link between hippocampal hyperactivity and clinical symptoms. we have not been able to do that very much. So we've not been able to tie hippocampal hyperactivity to illusions or hallucinations. It's even difficult to make a claim that hippocampal hyperactivity can explain the well-known relational memory deficits. While we can show that in our fMRI paradigms, we cannot really make the claim that these abnormal patterns of activity are the mechanisms of the well-known relational memory deficits. And it would be nice if we could get to that point, because then we would have a more consistent story of hyperactivity leading to cognitive deficits and clinical symptoms. All right, so I will finish my presentation saying that I'm still in place number 3. I don't know whether I will be there forever, but at least I've been there for a while now. And my curiosity has turned into some humble curiosity. All right. Thank you.
Thank you so much, Stefan. Can we just unmute ourselves and clap? It was fantastic. And as expected, I should say. Can just open up for questions and comments. Just unmute yourself and ideally.
Well, I have a question. I thought that was, as Brad Alger, I thought that was a wonderful talk. It was fascinating. And I thought every aspect of that was new to me. But I did have one question, though, which you slightly alluded to in one of your last slides. When you were looking at the hippocampal interneurons, you looked at the PV cells, the SOM cells, and some others, but you did not look at the CCK cells, the cholecystokinin cells.
Right.
The reason those are interesting, as you know, is that those are the only ones with the CB1 receptors.
Yeah.
This raises a kind of interesting question because cannabinoids will reduce the release of GABA from the CCK interneurons, right? And
Yep.
so what you might have then, I wonder about the link then between schizophrenia, and there's some developmental studies on the use of marijuana by young adults, on teenagers rather. And I wonder if you had a case where if your system is in fact slightly balanced towards disinhibition, and the CCK cells were providing some of the residual inhibition, if you then had them take cannabinoids, smoke marijuana, they would that would actually reduce the output of the CCK cells, which might, in the context of your hypothesis, as I understand it, tilt the system more towards disinhibition. So I wonder if you've ever, and that would also, by the way, suggest that another novel way of maybe an intervention, if there was anything to that story, then you might want to block cannabinoid receptors to prevent the disinhibition, and there's this drug that, as you know, was over-the-counter drug used for obesity control many years ago by Sanofi, and then it was withdrawn because it was kind of a depressant action on the CNS. I wonder if by any chance there might be some linkages there that would be interesting in your context.
All right. Thank you for that. Was more of a comment and question combined. And I tell you, this is very close to the discussion we had 3 years ago when we had to make a decision, what pharmacological modulation do we want to try to modulate hippocampal activity? And there were only 2 left after we did a very rigorous review. One was cannabidiol, and the other one was levetiracetam. And so there is an emerging story on this. Now, the Brits, particularly Phil McGuire and his group in London, have run with the cannabidiol story. In fact, they're doing a very large clinical trial of 300 at-risk, clinical high-risk individuals, where they use cannabidiol as a preventive strategy to decrease conversion to psychosis. And they use hippocampal hyperactivity as the biomarker and readout of that pharmacological intervention. So I want to share briefly, there's a paper that people should read. This is extremely exciting. It just came out a couple of weeks ago. I hope that people can see that. Cannabidiol modulates excitatory-inhibitory ratio to counter hippocampal hyperactivity. I saw this as, oh my God, we got it all wrong. We should have taken cannabidiol. They actually look at the GPR55 receptor, which they say is actually driving a lot of the CBD effects in the hippocampus. Now, this is a rodent model, but still, I'm very energized by that. I think there are now at least 2 different pathways. The one that we chose which is really targeting the excitation inhibition balance with an anti-epileptic, or with cannabidiol. Both of those are very promising strategies to normalize hippocampal hyperactivity. So that doesn't necessarily address your question. And to answer the CCK cells, in all of the anatomical work that we've done, we haven't looked at CCK. I can tell you it was not a major gene expression difference because we looked at gene expression as well. And it did not come out as one of the most promising targets. Somatostatin in palbamulin did. In fact, somatostatin was the #1 gene expression difference in this cohort. So.
Right. Well, that would fit with the idea that I'm putting forward. And by the way, cannabidiol doesn't affect CB1 receptors. So
yes,
that's an interesting kind of offshoot, but that's completely at right angles to the one that I'm suggesting, which is that I would predict in your model, and what I'm suggesting here is that CCK cells would not show any change. They would be among the ones hanging around to provide the residual inhibition. But when a cannabinoid agonist that would activate CB1 receptors is present, that would decrease the output from the CCK cells, and that might be the effect that would tend to tilt the system more towards disinhibition. And that is the idea that I was putting forward, to use not the cannabidiol, but the CB1 receptor antagonist.
Okay.
In your model, because that would then, I would suggest, tend to preserve the inhibitory effect of the CCK cells. Anyways.
Yeah, well, I did not pay enough attention to your question and jumped immediately from that initial one to cannabidiol. So
we had done a trial here with rimonabant about 10 years ago and found that it had positive effects on learning in schizophrenia.
Yeah, that would fit.
Yeah, this is Deanna. That's what I was going to say. We were about maybe halfway to 2/3 through the clinical trial when the European Union pulled rimonabant. If you go back and look at the data, the actual cases of depression and suicide were actually not significantly higher than the general population of because it was being tried in obese patients. Right. There were 2 or 3. Merck had one. There were 2 or 3 in trials at the time, and they were all sort of shelved. But I've had conversations in the recent years. These compounds are, sitting on the shelves of some of these pharma companies, and the data, these should be resurrected. I think they should be resurrected. And I think that we should all try to make attempts to work with companies to potentially do that. So I was just also encouraging you to see if it's possible to, get a hold of a CB1 antagonist.
Oh, I see that there are actually questions in the chat, which I very much appreciate. I typically don't pay attention to this, but I can address them. Stephanie here is asking whether of all of the 5 different parameters that contributed to the IHI metrics, whether there was one that is driving it more. Yes, and it was the criterion 3. And that was really the relative width of both the subiculum and the head of the hippocampus. And the ratio of that is remarkably changed in the incomplete hippocampal inversion. And that drove more of the effect than the others. And then Jim's question was about the conversion. So if there really is 1/3 of the early stage psychosis cohort that does not, over a 2-year period, does not progress to schizophrenia, what is their diagnosis at that time point, or even afterwards when we follow them? So I can address those questions. So we do not give them another diagnosis. We keep the diagnosis to be schizophreniform disorder. So they're not bipolar disorder with psychotic features or psychotic depression. They do meet criteria A for schizophrenia, and they do not have an affective psychosis. It is just that they are form fruits of schizophrenia. They might even have relapses, but they never reach the criterion where they actually have a extended period of criterion A for schizophrenia. If Will has some thoughts on that, because I think the DSM is somewhat silent on that and how to even think about. In fact, I remember I talked with Will and Rajiv about this years and years ago, how to even think about this. Do we have relapsing forms of schizophreniform disorder? But there's one other story, Jim, which is we are now following them 5 to 10 years into the illness, and we see a significant number of those converting later. So they are not staying away from schizophrenia. They could convert 5 years into the illness or even later. So that's somewhat similar to the Suffolk County data, where after 10 and 20 years, you get more and more of that consistent pattern of functional impairment and more pervasive psychotic symptoms, when they initially didn't show that, but over a longer period of time, there are more progressions or conversions.
Thank you.
Stephen, I would only add the comment that the studies that over time have shown that in the neighborhood of 20% of people with schizophrenia seem to have a good prognosis. And I think those are mainly based on things that are followed for a few years, but not for a lifetime. And I bet the general practice is that they just stay with the diagnosis of schizophrenia and remind us all that not all people with schizophrenia are on a chronic course to begin with.
Yeah, and so in my slide, I captured that also with that second quote. Once you've progressed to schizophrenia, you meet the criteria at any point. Within that, you can then still follow them over a long period of time, and you find some 10 to 20% who have better social functioning and have better outcomes as defined either by clinical symptoms or by social functioning. But my interest really in the schizophreniform disorder group was that this is upstream, much closer to the early stages of the illness, where we do have quite a bit of heterogeneity. And again, we've had this debate about brief psychotic disorder, cycloid psychosis, polymorph presentations, schizophreniform disorder with and without good prognostic features. How can we separate them? There's tons of literature on this that I had not really paid that much attention to early on. But now have become very interested in that, because it seems that what we are trying to do in early stages of psychosis is very similar to what the ultra high risk research community wants to do, which is showing what predicts the conversion from clinical high risk to psychosis. We then take it one step further. The psychosis has emerged. The person meets criteria A for schizophrenia, but then we have variable degrees of impairment following that over the first 6 months or even 2 years. And all we are trying to do is capture that heterogeneity, and found there were some striking differences between that fit more into the traditional schizophrenia cohort and those that were not.
I'm looking around. We could go on for a long time, right, Stefan? And that's the problem with not being here in person. We could have gone on for a lot. You told us before, some of us were on already, that you have to see a patient in a few minutes. So yeah, maybe what we should do is just stop here, and we of course be in contact, and everybody knows who you are, where you are, and vice versa. So let me just thank you one more time for doing this and for your absolutely wonderful, not only the work, but also the presentation. It makes us think a lot, and we very much look forward to our next interactions.
Well, thank you very much. Can you just tell me briefly, is that a meeting that happens like every month or more frequently? And kind of
yeah, so we have monthly, so this is part of the Conte Center, and we have usually 8 to 10 monthly meetings in a year.
Okay.
So in the summer we usually don't, but yeah, so we have a meeting in the morning like we had today, where we talk about our own internal progress, and then we have an external speaker who is usually world famous, like in this case.
I'm asking because I watched Joe running the Conte Center, and I'm also aware of some other Conte centers. So it's really interesting to see how you're managing this, that you are also interested in hearing from others who might do things that are, quite different, using methods that are different, and just using that as a group to kind of brainstorm about what you want to do. You've been now doing the Conte Center for 7 years, or?
We're in year 9, actually.
9 years, okay, wow.
So we think of the future. So we can talk about this also. The hippocampus is certainly not something we are focusing on, because we don't have internal people to that, but we definitely are open, and other people of course in Conte Centers too, to work with people outside our own institution. I mean, if there's somebody can do something better, much better than we can, why not, right? And the NIMH is fine with that. So no, you're absolutely right, and that's exactly why we are doing this, and we're learning. I mean,
And if Diana is still there, are you still doing your trial of, I think it's a gluten-free diet, is that correct?
Yes, we are. So we're in the last year of the gluten-free diet, and it targets people who have antibodies to gliadin. So we're recruiting people who have a high immune response. We find that those people who have this immune response also have pretty high inflammatory markers, and then sort of with our MRS data looking at brain markers that proxy inflammation, they also have correlations with the higher the antibodies, the higher this sort of potential brain inflammation. So this subgroup of people is sort of been our target, but yes, we're hoping to finish up this year sometime.
Yeah, I'm asking because I know a couple of years ago I contacted you because I had 1 or 2 patients who might qualify for that, but I could never convince them to do something like that. So yeah.
Thank you. I appreciate you thinking about it.
All right. Well,
Stefan, wait a bit. Give me 30 seconds, because everybody who is still on will say, why didn't I ask my favorite question? And some people stopped talking to me after I did this 20 years ago. Neuroleptics. You did not mention dopamine or neuroleptic treatment at all. Are you thinking, I'm sorry, this is, of course, you can give a long answer. Just briefly, is this part of your thinking about neuroleptics in the hippocampus? Just very briefly, and then we can continue later.
yes. Well, if Tony Grace had given this talk, he would have told you exactly what I told you, but he would have ended, and all of that downstream leads to a dysregulation of dopamine release, because the hippocampus is a major driver of dopaminergic firing or release of dopamine into target regions. So, of course, he's done in John Listman's paper that I cited, the Transit Neuroscience paper, was very much based on the Listman-Grace hypothesis that really whether that's the hypersalient syndrome as a kind of a psychological construct, or the pharmacological differences when it comes to dopamine receptors, that they all can be tied to a hippocampal mechanism.
Yeah, exactly.
So
By the way,
I just didn't make that case because that's not really where I'm looking, and I've not really done PET imaging to say something about. There are some people who've tried to look at dopamine functioning with fMRI, but I've not done that. I tell you, however, that I am painfully aware of how important neuroleptic treatment or treatment with antipsychotic medication is, because it modulates some of those markers that we actually are studying. It does not have a significant effect on overall hippocampal volume. And based on the data that I showed you, the DC hippocampal volume difference might have a neurodevelopmental etiology and might already be present early on. I'm not surprised that our striking hippocampal volume differences that we always find are not simply a drug effect. However, they have a huge impact on some of our functional imaging markers.
That's exactly my question, Stefan.
And that's why I couldn't show you the data from our clinical trial, because they were all medicated patients. Yeah. And we've now learned that arterial spin labeling, which is considered to be the new gold standard, because people don't do O15 PET anymore, and very few people do CBV. So they really want to look at arterial spin labeling, because it's also an absolute measure of blood flow. People are really pioneering this and saying that that is what we have done it for years, and there are huge medication effects, particularly antipsychotics, and that worries me. CBV does not seem to have the same vulnerability, and so we tried various methods and now know kind of which method is better suited to study hippocampal hyperactivity in a person who is medicated, because most of our patients are medicated.
Okay, thank you.
The gradation that you showed between early and late psychosis in terms of hippocampal volumes, those are, I mean, most of the evidence points to that being as a consequence of chronicity and not chronicity of drug treatment.
Right. And again, we looked at this over a 2-year period. We are now looking at the same cohort 5 to 10 years into the illness. We don't find any hippocampal volume differences over a 2-year period, scanning them every 6 months, and also found no correlation of that with medication status.
Okay, Stefan, we should let you go. I'm sorry to continue, but there's so many, we could again talk for a long time. I want to thank everybody in the audience also to be part of that. Everybody was very, as you noticed, very interested in this, and we'll all follow up, okay?
All right.
Take good care.
you.
Thank you so
Thank
much.
you.
Bye-bye.
Bye-bye.